Hello students, I am going to describe the microbial diversity. Basically, the microbial diversity is a vast chapter and it is impossible to finish this chapter in a single class. Basically, the microbial diversity is the classification of microbes, which is based on different methods and different techniques and different parameters. So I, as this class is a limited time period class, I am trying to describe this on the main points, on the basis of main points by which the microbial classifications are done. So microbial diversity considers the vast array of microorganisms, the smallest form of life, which exists everywhere. It is the smallest form of life in which consists of there is a DNA or genetic material, or there are some functional units like proteins. The three primary groups of microorganisms are bacteria, archaea, and eukarya, and this is shown in the figure. These are the domains of the unrooted life tree, and the bacteria and archaea are the prokaryotes, and the eukaryotes are the one which is different from these prokaryotes. The prokaryotic organism contains the genetic material that is in a single chromosome. In eukaryotes, the most of the genomes are held in multiple chromosomes, and in the prokaryotic domain bacteria, over 11,000 species are identified till date, and their identification was on the basis of the shape, metabolic activity, gram staining techniques, and genetic identification of DNA and RNA sequences. The archaea is the another domain where around 500 species are there. These are divided into two phyla. One is Euarchaeota and Crinarchaeota. Now, what is microbe? Microbe is the microscopic organism, and this is the smallest form of life. This can be classified into two categories. One is cellular, another one is acellular. The cellular category is further divided into prokaryotic and eukaryotic group. In the prokaryotic group, the bacteria, archaea are there, and eukaryotic group, some microscopic algae, protists, and fungi are there. In the acellular group, there are the viruses or virusoids or some prions. I previously told that the bacteria, eukarya, and archaea are classified on the basis of several parameters, but most importantly, the molecular basis of molecular parameter, which is based on the DNA and RNA sequencing, is very much important. Basically, the ribosomal RNA sequencing is used for the phylogenetic analysis and classification among the form of lives. Here, the rRNA sequence differs from each other, and on the basis of this difference, the taxa are generated, where these domains are separated on the basis of rRNA sequencing. So bacterial classification on the basis of phenotypic and genotypic classification. In the phenotypic classification, we can classify the bacteria on the basis of the microscopic and macroscopic morphology, the biotyping, stereotyping, and antibiogram patterns, and the phage typing. In the genotypic classification of bacteria, the guanine cytosine ratio, DNA hybridization, nucleic acid sequence analysis, plasmid analysis, ribotyping, and chromosomal DNA fragment analysis was done for this classification. According to a proposal by Carl Woese, the world of living things is classified in the three domains: bacteria, archaea, and eukarya. In this system, each domain is subdivided into kingdoms. In case of bacteria, there are further classified into on the basis of the metabolism, the heterotrophic bacteria and the photosynthetic bacteria. The heterotrophic bacteria are the bacteria that are pathogenic in nature, and they often cause the disease, human disease or any type of animal disease. And the photosynthetic bacteria like cyanobacteria, those are non-pathogenic. They can be classified, the bacteria can be classified on the basis of the reproduction and binary transfer fission. They can also be classified on the basis of the parasitism and the shape of their shape, and also they can be classified on the basis of the presence of cell wall. When the bacteria which is without rigid cell wall, they can be classified under the mycoplasma, and they are known as the unique bacteria. When the shape of the bacteria are the main concern, we found that there are three types of shapes. One is cocci, the round shape. Another one is bacilli, a rod shape. And in the other shape, where the various shapes of bacteria can be found, like vibrio or comet shape, some spirulina or spiral shapes, and the spirochetes, like thread-like structure, are found. Now, the cell wall, now come to the point of cell wall of archaea. The archaea is the primitive prokaryotic organism which is divided from bacteria, which is basically separated from bacteria on the basis of their cell wall structure. Basically, archaea cell wall differ from bacteria cell wall in their chemical composition, and they lack peptidoglycans. The archaea builds the same structure as other organisms, but they are the form different chemicals compounds. The archaeal cell wall is assembled from the surface layer proteins called S layers. Likewise, archaea do not produce walls of cellulose, as the cellulose is found in plants, and it devoid of chitin, which is found in fungi. The most striking chemical difference between archaea and other living things lie in the cell membrane. There are four fundamental differences between the archaeal membrane and those of the other cells. First one, the chirality of glycerol, the ether linkage, the isoprenoid chain, and the branching of side chain. First, I will describe the chirality center of the glycerol, which is depicted here, and there is a phytanyl side chain which is present there, and the phospholipid is branched in case of archaea. And another thing is, in archaeal cell wall, there is we can found the CO bond CH2. This is the ether linkage, O double bond. The O is the middle portion, oxygen is in the middle portion, is known as the ether linkage, and whether we can find the ester linkage in other bacteria or eukarya. Another difference is that the phospholipid bilayer form is continuous, whereas in phospholipid bilayer form of bacteria and eukarya are separated, and this is not continuous. So the rigidity of the archaeal cell wall is much more than the bacteria or eukarya. Now come to the acellular part of the microbial diversity, which is virus, and mainly the tumor virus is important. We choose tumor virus for this topic because this virus can consisting of the two types of viruses, like RNA virus and DNA virus both. These are basically the oncovirus that cause cancer, and the term oncoRNA virus was used to denote their RNA virus origin, but nowadays we can find that there are some DNA oncoviruses are there, and these are now classified into tumor virus. They are known as tumor virus now. The vast majority of human and animal viruses do not cause cancer, probably because the long-standing co-evolution between the virus and their host. The oncovirus have been important not only in epidemiology but also in investigation of cell cycle control mechanism such as retinoblastoma protein. The basic latent life cycle of a tumor virus is depicted in this picture. The virus first infects the cell, and its integration happens. After that, the cell is transformed. Some virus-specific protein expressed, and no mature virus is there. The viral structural protein not expressed, and changes in the proteins of host cell is called the transformation. Sometimes latency may terminate, and the cell must be infected by the complete virus. Now come to the next slide. where the DNA mechanism of DNA of tumor virus and RNA tumor virus are depicted. Basically, when the DNA tumor virus attack on the host cell, the host RNA polymerase 2 converts this DNA into the mRNA, and the host enzyme converts this mRNA into proteins. And ultimately, the virus will form, and or transformation happens, which generally develop tumors. In case of RNA tumor virus, both the mechanism is similar to the DNA tumor virus, but here the viral RNA genome is converted with the reverse transcriptase enzyme, which is from the virus encoded, and it is converted into the viral DNA genome, and this DNA genome integrated into the host DNA and use the host DNA RNA synthesis mechanism, and ultimately it gives rise to the protein expression, and by this process the tumor will develop. Now come to the another example, which is Escherichia coli. That is the main very important model organism in case of microbiology and biotechnology. The Escherichia coli is the gram-negative facultative anaerobic bacteria. It is rod-shaped and coliform bacteria of genus Escherichia that is commonly found in the lower intestine of warm-blooded organisms like human. Most E. coli strains are harmless, but some stereotypes can cause serious food poisoning in their host and are occasionally responsible for food contamination incidents. The harmless strains are part of the normal microbacteria of the gut, and they can benefit their host by the producing vitamin K12, which helps blood to clot and preventing colonization of the intestine with pathogenic bacteria. They having a symbiotic relationship with the human gut in the human gut, and they stay here, and in the exchange they produce vitamin K12 inside the intestine. E. coli is expelled into the environment within the fecal matter, and the bacterium grows massively in the fresh fecal matter under aerobic condition for 3 days, but its number declines slowly afterwards. The yeasts are the eukaryotic example of microbial diversity. It is the eukaryotic single-celled microorganism classified as members of the fungus kingdom. The first yeast originally originated hundreds of millions years ago, and at least 500, 1500 species are currently recognized. They estimated to constitute 1% of the all described fungal species. Most yeast reproduce asexually by mitosis, and many do so by the asymmetric division process known as the budding. As with their single cell growth habit, yeast can be constructed with molds which grow hypha, and fungal species that can take both these forms depending on the temperature or other condition are called dimorphic fungi, and yeast is the dimorphic fungi. The yeast species Saccharomyces cerevisiae converts the carbohydrate to carbon dioxide and alcohol. It is called the process of fermentation. These products of this reaction have been used in baking and production of alcoholic beverage for thousands of years, and this is the main organism used in the process of fermentation in industrial biotechnology. So students, please go through these slides, and I think this will be helpful for your exam. Thank you.